Welcome dear students. In this video we are going to learn about JAK-STAT signaling pathway. At first I want to mention that this JAK-STAT pathway and this RTK, that means receptor tyrosine kinase pathway, is very similar. They perform the functions very similar way, but there is a basic difference you have to understand. In case of RTK pathway, that means receptor tyrosine kinase pathway, the receptor that actually binds with the ligand have intrinsic kinase activity. That means, let's suppose if this is the RTK receptor, this receptor has a kinase domain in the cytosolic region. This is the plasma membrane, this is the receptor. It has a kinase domain that actually has an intrinsic property of kinase activity. And in case of RTK and JAK-STAT pathway, both the receptor acts as a tyrosine kinase. So both are actually tyrosine kinase, but the difference is, in case of RTK, the RTK receptor, that means receptor tyrosine kinase pathway, the receptor has intrinsic property of kinase activity. Okay, but in case of JAK-STAT pathway, the receptor don't have this type of domain. They don't have the intrinsic property of kinase activity. They associate with a kinase protein or enzyme that is called JAK. JAK means just another kinase. When scientists discovered JAK protein, they named this protein just another kinase. So I will talk about in this class about the JAK-STAT pathway. Before I start this signaling pathway, I have mentioned a basic fact about the difference between JAK-STAT pathway and RTK, because both pathway works similarly. They both activate tyrosine kinase, but in case of RTK pathway, the receptor has intrinsic property of kinase activity, tyrosine kinase activity, but in case of JAK-STAT pathway, the receptor actually associated with a kinase called JAK. So it is also known as receptor associated kinase signaling and is called receptor tyrosine kinase. Okay, now first thing is you have to remember the ligands that actually activate JAK-STAT pathway. So the ligands is very important. The example of ligands are, you must remember all the cytokines activate JAK-STAT pathway. Cytokines are some molecules that are produced by immune cells. You can divide the cytokines into two main ways called interleukins. We call in short IL. Then there is IL-1, 2, 4, 5, 6, look like this. And it actually there are so many different type of IL interleukins: 35, 19, 15, 21, 22, 17. There are so many interleukins are present. It's called IL. IL means interleukin. Another type of cytokine we call it interferon. These interferons actually perform different functions. They actually helps in the viral resistance. So cytokines are actually messenger between different immune cells, and interferons act as to actually create the viral resistance in our body. So cytokine is one example of this ligand. Okay, another is some hormones like growth hormone, prolactin that helps in the milk secretion, then GCSF. GCSF means granulocyte colony stimulating factor. CSF, colony stimulating factor, granulocyte colony stimulating factor. Then thrombopoietin. This thrombopoietin helps in the production of platelets, create platelets. Another is called erythropoietin. In short, we call EPO. It helps in the production of RBC, red blood cell. So these are the major ligands of this pathway. Then you have to remember the receptor. So these are the ligand, and what are the receptor? There are so many receptors are present for cytokines. There are some receptor present for growth hormone, prolactin, GCSF, thrombopoietin, erythropoietin. One information I want to share, that these molecules share a very common structure this growth hormone, prolactin, GCSF, thrombopoietin, erythropoietin. That means they are evolutionarily very conserved. So when they bind with the receptor, the structure of the receptor is also very similar. But the cytokine receptor is different. Okay, so the receptor could be different, but the thing is here, they always form homodimer. They always form dimer. That could be homo, that means homodimer. Or in case of this interleukin and interferon, they can form heterodimer. In case of growth hormone, prolactin, GCSF, thrombopoietin and erythropoietin, they form the homodimer receptor. But in case of interleukin and interferons, they form heterodimer. Okay, this is another fact. And another important fact about this signaling pathway, that there is a kinase that associates with this receptor, that is called just another kinase. Just another kinase. This kinase actually has a tyrosine kinase activity, so it can phosphorylate particularly on tyrosine amino acid. Then this JAK protein can activate directly a transcription factor called STAT. There are different types of STAT also present: STAT1, STAT2, STAT3, STAT4, STAT5, STAT6. There are so many STAT proteins are there. So they directly activate this STAT protein called transcription factor. Another ways are there that can be activated, like they activate a protein or actually associate with a protein called GRB2, or you can write GRB2. This GRB2 actually activate, the RAS MAP kinase pathway. Another way that JAK actually can activate a protein called PLC gamma. This RTK pathway can also activate this PLC gamma. So you can see here is a interconnectedness of this signaling pathway. JAK pathway can activate PLC gamma. RTK pathway can also activate PLC gamma. And how this JAK protein activates these other different proteins, I will show you. At first you remember that JAK protein can actually diversify. It can activate JAK-STAT, so the name implies JAK-STAT. But it can also associate with a protein called GRB2 that actually activates the RAS MAP kinase pathway, and can activate PLC gamma that actually associates with RTK pathway. And ultimately PLC gamma actually activates or increases the concentration of the calcium. Okay, another protein, like I can write here GRB2, PLC gamma. Okay, so these are the proteins that can be activated by JAK actually a kinase. Now, yes, another protein is that activate the mTOR pathway, that is called PI3 kinase, that activate PKB. This PKB protein can activate the mTOR pathway. Can you see here that this JAK-STAT pathway, JAK pathway actually can activate different proteins. So they are actually interrelated. Now I will show you how this signaling process occurs. This is very important. Another thing I want to mention, that the information I am sharing with you is very limited, because I also refer for the cell biology and molecular biology the book of Lodis, 8 editions, cell or molecular biology Lodis, and there is Bruce Albert. Then you can actually find the information from different other cell biology book, like Lewin cells, another is Pollard, there is Karp cells and Cooper. I always suggest these 6 books for your CSIR preparations. So if you want to prepare yourself for the CSIR NET, you must actually collect the information from the 6 book. Okay, now I will show you the reaction or the pathway, how this occurs. JAK-STAT pathway. Okay, so let's suppose this is a plasma membrane where the receptors are there. The receptor, these are the receptor. Okay, this is the JAK-STAT pathway receptor. It could be the receptor for cytokine or growth hormone, prolactin, GCSF, or it could be the receptor for thrombopoietin or erythropoietin, whatever may be. Actually, when this receptor do not bind with the ligand, they remains as a monomer. when they bind with the ligand, let's suppose this can be any ligand, they actually form the dimer. But another the exception is that the EPO, that means erythropoietin receptor, remains always as a dimer, whether there is EPO or not. EPO receptor, erythropoietin receptor, always remain dimer when there is no erythropoietin or there is erythropoietin. But other cases, they remains as monomer. When ligands bind, they form dimer. And when this dimerization occur, one important thing occur, that is, in all the way when this receptor is active or inactive, there is a protein that associates with this receptor called JAK. Okay, specifically JAK2. I want to mention here that all the hormone like growth hormone, prolactin, thrombopoietin, erythropoietin or GCSF, they associate this receptor associated with JAK2. Now, when this ligand bind, this receptor actually come close to each other, make the homodimer. The receptor actually make the homodimer, and this homodimer receptor actually perform a very important function. They actually helps the JAK protein to phosphorylate each other. That means this JAK protein phosphorylate its tyrosine, particularly a position called activation loop. The JAK protein has an activation loop. When this activation loop gets phosphorylated, the JAK proteins actually become active. This is the way how JAK protein actually get activated. You have to understand this. When there is monomer of the receptor, JAK protein is not active. The activation loop is not phosphorylated. When ligand bind, these two receptor come close, and then JAK protein actually phosphorylate the first tyrosine that is present in the activation loop. When this JAK protein gets phosphorylated in this activation loop, both these JAK protein in the activation loop, they then phosphorylate the receptor on particular tyrosine residue. Always the tyrosine residue. They phosphorylate the tyrosine residue. On different tyrosine residue of the receptor. Now, the transcription factor that I have called that STAT, this protein has a domain called Sh2. This protein has a domain called Sh2 domain, through which it can bind with this phosphate, because Sh2 means Src homology to Src. Src. This is a very important protein. It's actually found in case of chicken. This is actually a proto-oncogene. When it becomes oncogene, it actually causes sarcoma. So from that name, it is called Src. So this is called Src homology 2. You know, there are different type of domains are there that can bind with phosphotyrosine phosphate. So this means phosphotyrosine. When tyrosine gets phosphorylated, there is some domain that can bind with this phosphate group. One is called SH2 group, and this SH3 group, like PTB group, they can bind with phosphotyrosine. Okay, so this STAT protein has a SH2 domain that binds with this phosphate group. So STAT protein actually get attached with this receptor STAT through the help of SH2 domain. SH2, you must remember this, Src homology 2, because they have this domain through which it actually interact with this phosphate group. Then this JAK protein has the opportunity to phosphorylate this STAT protein. So this JAK protein phosphorylate this STAT protein at particular site. When STAT protein gets phosphorylated, they actually dissociate from the receptor, and the phosphorylation actually occurs in the C terminus of the STAT protein. So when this STAT protein gets phosphorylated, they actually make a dimerization or dimer, STAT dimer. This is always homodimer. You remember, STAT protein always make homodimer. The receptor can be homodimer or heterodimer, but the STAT protein always makes homodimer. That means STAT5, STAT5, STAT1, STAT2, STAT1. So when they phosphorylate, they dissociate and they form dimer. And the thing is, when they make dimer, they actually expose a region called NLS. At first it was not exposed. This is called NLS, nuclear localization signals. In my previous video TJ beta, I have told that in case of SMAT proteins, also the NLS domain is there. That is called nuclear localization signals. That means this domain can be recognized by a protein called importin, and importin actually helps in the import of this protein into the nucleus. And in the nucleus there is a DNA. When this DNA and this STAT protein binds with this DNA, they help in the transcription of the gene, particular gene. In case of immune cells, there are different genes that could be activated by the cytokine signaling, and in case of prolactin signaling, they help in the production of milk. In case of erythropoietin, they help in the production of a protein called BCL-XL. That is actually anti-apoptotic protein that prevents apoptosis, so erythropoietins or RBC survive. So by this way they can actually control different genes. Another important thing I want to mention, that this signaling pathway inhibits itself, because this STAT protein binds with another protein that actually helps in the transcription of another protein called SOCS. SOCS means suppressor of cytokine signaling. I will show you how this occurs, because this is the ending of this signaling. Because I have mentioned in my first basic concept of cell signaling that when there any signaling starts, it should be stopped. Okay, so how this JAK-STAT pathway actually stop? This is the pathway. Now how it stop? There is a two way of this stoppage. There is a two way. The first way is by a protein phosphatase called SHP1, and another way is called SOCS, that means suppressor of cytokine signaling. I will show you how these two proteins work. I have mentioned that when this is the plasma membrane, when ligand bind with the receptor, that is the JAK-STAT receptor, then JAK, that means kinase, actually phosphorylate the receptor or on particular tyrosines. This is the JAK protein. Let's suppose this is the JAK protein and this phosphorylates this tyrosine residue. When this receptor gets phosphorylated, this phosphatase called SHP1 has two domains. Let's suppose this is the phosphatase domain. This is the catalytic domain, C domain, catalytic domain. But this domain is actually masked. This domain is actually masked by another region that is called Sh2 domain. I have mentioned previously that STAT protein contains Sh2 domain. Sh2 protein domain can bind with phosphotyrosine. So when the receptor gets phosphorylated, this SHP1 gets activated. When the receptor is not phosphorylated, then this protein remains as it is, inactive in condition, it is off in state. This is the catalytic domain. You have to understand this. This is SHP, that means protein phosphatase. When this signaling is not occurring, there is no phosphate group in this receptor, then this SHP protein remains as this structure. This Sh2 domain actually masks the protein phosphatase domain so that it cannot perform its function. But when this ligand binds this receptor activators, then a transcription actually occurs. After a few times, this Sh2 domain binds with this receptor, then the phosphatase domain becomes open or active, because now the Sh2 domain binds with this region, so it actually free the phosphatase domain. Now this phosphatase domain can remove the phosphate group of the receptor so that no longer STAT proteins can bind. This is a very interesting pathway. or a mechanism of negative feedback loop. And is called SOCS. How SOCS work? SOCS works also by this way. SOCS protein also have Sh2 domain. This is very interesting fact. This protein also has Sh2 domain, but it is also has a box called SOCS box. This SOCS box can recruit E3 ubiquitin ligase. You know that ubiquitin ligase can ligate ubiquitin, and when a protein gets polyubiquitinated, you have to understand this. In case of monoubiquitinations, when a protein gets monoubiquitinated, then it actually performs different functions. I will make a video on ubiquitinations because it is very important. Monoubiquitinations and polyubiquitinations is very different in mode of actions. But when polyubiquitinations occur, that means many ubiquitinated ubiquitin actually gets ligated with the protein. This is called polyubiquitinations. Then the protein actually destined to the proteasomal degradation. You have to understand this. This protein gets degraded. The same way is here. When E3 ubiquitin ligates actually polyubiquitinate this receptor actually get degraded. Another thing, this SOCS protein can bind with the JAK protein because JAK protein is also phosphorylated. I have mentioned activation loop is actually phosphorylated. So the JAK protein also degraded. So this is all about the JAK-STAT pathway. Hope this video will help you. If you have any doubt, you can write me in the comment section. Thank you for watching and always keep learning.